Good afternoon, everyone. My name is Rana Reis, and it gives me great pleasure to introduce our speaker today, Dr. Suvendra Battacharyya, who's a professor at the Department of Pharmacology and Experimental Neuroscience at University of Nebraska Medical Center. Dr. Battacharyya possesses a diverse background in molecular biology and biochemistry, with a specialized focus on RNA biology and cell biology related to miRNA-mediated processes. His research primarily investigates the mechanisms underlying gene expression changes mediated by mRNAs, and notably, his laboratory is at the forefront of global research on understanding the mechanisms involved in the export of mRNA through exosomes and extracellular vesicles. And I think that's what his talk is about today. So without any delay, I'd like Dr. Battacharyya to start his talk. Thank you.
Thank you, Dr. Rice, for the kind introductions, and also to the community for inviting me for this talk, and Cassie also for arranging the whole things. And I just start sharing my screen, so I will start with that. Yeah, I think we are all set. So I'd like to talk today on a few things that are all related to the extracellular vesicles. And I will comment on and present to you about my findings on how the microRNAs get packaged into the extracellular vesicles, get exported out, and how they get transported across the matrix of the tissue, and how they're taken to the target cells for the functions. So, with that, I'm going to introduce to the first slide. So that is about how the microRNA-mediated gene repression happens. So microRNA, you all know, are very tiny molecules or tiny RNA that are processed from the prime microRNA by the DORSA-DGCR8 protein complex. And these DORSA-DGCR8 protein complex act on the pre-microRNAs and to produce the pre-microRNAs from the prime microRNAs, and this is get exported into the cytoplasm and matured there by the enzyme complex DISA-TRBP. And this DISA-TRBP complex produce the mature microRNA of 22 nucleotide long from this the hairpin structures pre-microRNA. And this is then incorporated into the Argonaute protein complex to form the Argonaute microRNA complex that find the targets and bind the mRNA with complementarities, and they induce translation depression or degradations. There are thousands of microRNA operates in humans, and almost half of our genes are under microRNA regulations. MicroRNA regulate different stages of development, differentiation, and apoptosis. Their expression is also getting dysregulated in human diseases, and microRNA also showed developmental and tissue-specific expression pattern. For example, there are microRNA specifically expressed in brain, liver, or muscle. So when we talk about these kind of microRNA-mediated depressions, so we always ask about the reversibility of the process, and that was one paper we published during my postdoc time to show that this process can be reversed, because microRNA messages are also stored in kind of cytoplasmic structure called RNA processing bodies, or PBs or P-bodies, and can be reversed for the action of certain RNA binding proteins like HUR. Under amino acid starvation conditions, this HUR protein shuttles from the nucleus to the cytoplasm in human hepatic cells and de-repress the microRNA-mediated depression by binding the messenger RNAs and replace the microRNP from the target messenger RNA. So that was one of the first reports to show that there are other RNA binding factors that can positively or negatively influence the microRNA activity. And in that context, HUR is one of the good candidates to explore that thing. But this external and internal cues-driven recruitment of these RNA binding proteins to the UTRs may cause a difference in the microRNA activity by positively or negatively regulating their activities. So with that, I like to also to show that this modulation of microRNA activity can happen in the mammalian cells. So that are the different four line of research that we are involved in. So one of the that's why we are talking about how the microRNA getting deactivated during the differentiation of neuronal cells. We work on the mechanism and action of this process. We also work on how the alteration of microRNA function happens during the pathogen-inhibited macrophage. And in that case, we use a model, Leishmania donovan is a parasites that infect the macrophage and replicate within. So how they cause the changes in the microRNA machineries and how the microRNA is useful to combat the disease is a research area that we also explore. We also want to know the roles of different subcellular organelles like endoplasmic reticulum and mitochondria in the process of microRNA biogenesis control and activity in different cell types, particularly in the immune cells and in cancer cells. And today I'll focus on the regulation of intercellular microRNA transport process that happens in immune and cancer cells to affect their activity, biological activity, and that is the work I'm going to present today. So it all started in very simple manner when I started my lab in Indian Institute of Chemical Biology, Kolkata, India in 2008 with a Human Frontiers Fund to check how the microRNA-mediated replication process is influenced by the extracellular context or the matrix. So we started very simple. We grow the cells from low density culture to the high density culture, in this case, HeLa cells. And you can see that with this change in the density of the cell culture, you can see that there is a change in the microRNA level that happens in the HOH7 cell or in HeLa cells with the density. The microRNA 1 to 2 or lead 7 gate goes up with the density of the cells. And we find that this actually happens for different microRNAs, and it not due to the increased biogenesis, but due to the higher stability that happens during this process. So we also find that intercellular transfer of microRNA happens through exosomes or extracellular vesicles. So exosomes are formed in a very complicated manner through the endocytotic process, where the endosomes mature to the late endosomes and multivesicular bodies. And these multivesicular bodies carrying the microRNA can be fused to the plasma membrane to release the content, and then we call them exosomes, but more popularly they are now known as extracellular vesicles. We'll try to label them as extracellular vesicles from now onwards. So microRNA activity regulation could be a very effective way of regulating this process. And the question we had at that time, does reduced extracellular export can contribute to the increased microRNA level that we have observed in the growth retarded cells? So we have done the experiment here to measure the microRNA level in the exosomes or extracellular vesicles to find that both in MD, MD231 cells or HeLa cells, the level of microRNA goes actually exosomal export goes down or when there is a confluence increase of the cells. So no change of exosomal, non-exosomal pool of microRNA occurred. And if you use a blocker of the exosomal export process, like for example, GW4869, one can really block this kind of increase of microRNA level in the exosomes. So that suggests that the exosomal export process is contributing to the increased microRNA level we observed. And we published the paper in MBOC. My first PhD student published that work to suggest that actually there is a polysomal arrest of the microRNP happens during the high confluence state that allow the restricted microRNA export occurs in the growth retarded cells or senescent cells. So then we went on to explore the more mechanistic detail of the process to find out is actually the mitochondrial activity control the mTORC1 activity that ultimately leads to the compartmentalization defect of EIF4 to the ER, and that is regulating the export process of microRNA because of the retarded recruitment of EIF4 under the growth arrested conditions, because of the low mTORC1 activity and low mitochondrial activity that allow the microRNP to be retained on the polysomes and no low export occurs. But in the proliferating cells or in cancer cells, this process is actually the reverse due to high mTORC1 activity and more EIF4 recruitment. The export process is higher. So that paper was published in Journal of Cell Science. So we see that exosomal export could be a very interesting way of regulating microRNA export. There are different processes that one can think about that are regulated by extracellular vesicles, and they have different kinds of cargoes from nucleic acid to proteins to amino acids, some metabolites, and in their membrane they have specific protein present like CD9, CD63, or CD81. They're important proteins, and they are present on the membrane of these single membrane vesicles. But the problem is that how the exosomal or EV cargoes are sorted. So I want to highlight the importance of these extracellular vesicles in the context of HIV infections, because I am sure most of the audience in this community are willing to know about the exosomal export and HIV infection process. And we have the evidence to suggest that the EVs or extracellular vesicles secreted by the infected cells may have a positive role to play to promote the HIV infections. It can contain the viral components. On the other hand, it also there are exosomes released by the other kind of cells surrounding the body fluid generation, like semen and vaginal fluid. They also have the EVs that can combat the infection process. So that's summarized nicely in a review. So when you go into the more details about the pro-HIV actions of the factors that carried by the extracellular vesicles, we can find that they can affect in different stages. They can have two different kinds of cargoes, like HIV cargoes and cellular EV cargoes. And then you can see that they can have impairing the antigen recognition process by affecting the CD4, by delivering the CD4 and affecting the CD4 expressions. And also it affects the deterring of IgA or IgG and enhancing the immunosuppressive cells. And also it affects the viral tropism by affecting the level of CXCR4 or CCR5 or TAT or ENV into the system by delivering them into the target cells. And also it can deliver certain microRNAs and viral microRNAs also, and cytokines also, to induce the production of pro-inflammatory cytokines there. On the other hand, there are also evidence to suggest that anti-HIV actions that are also possible with certain EVs that can carry specific microRNA like microRNA 28, 29, 125B that inhibit the translation of some of the viral proteins, or it can also affect some of the viral transcription process or stability of this transcript by affecting the level of microRNA 17, 20, or 29B. So one can think about how can we use this EV and its cargo to restrict the HIV infections. To know that, we have to understand how the microRNA are exported via this exosomal content or EV content. So what are the extracellular factors that control this kind of microRNA trafficking between cells that we want to address? And we also want to address what is the mechanism of this kind of selective export of microRNA via exosomes. That was the initial experiment we have planned. As I mentioned to you, this HUR is all interesting protein that uncouple the microRNPs from the target mRNAs by replacing them, and they oligomerize on the microRNP to form the 3' UTR, some kind of polymer on it to affect the microRNA repression process. But what happened to this replaced microRNP? That was the questions. And when we use the start cells to isolate exosome to find that this miR122 is specifically get exported into the extracellular component or exosomal component to show an increase in the exosome under the starvation conditions, which is not happening with the other microRNA. And it also affects the target gene expression in the target cell. So microRNA are getting exported out. And when you use the HUR siRNA to reduce the microRNA level, HUR level into the cells, we find that the relative level of cellular microRNA level increases, as well as the exosomal microRNA level decreases. That suggests the HUR is actually necessary for this kind of extracellular export, and it happens in the other context also. And starvation and HUR-driven export of microRNA also happens in the animal liver that we have tested. And HUR depletion in amino acid starved HUS7 cells actually prevent the exosomal export of microRNA and upregulate the cellular miR122 level and downregulate miR122 target gene expression. That is evident here. In the other time, we find there is a binding of microRNA happens with the HUR also under the starvation condition. So that suggests HUR not only replaces the microRNA from the target genes, but also it binds the microRNA and probably play a role in the export process. So starvation and HUR-driven export of miR122 control also the stress response that happened during the amino acid starvation kind of stress in hepatic cells. And HUR-mediated export also happens in the other cell types. Not only it's exclusive for the hepatic cells, but the other cell types also happens. So how HUR facilitated the export process of microRNA? So we explored the mechanism in detail, and I like to summarize the finding here in this paper we published in EMBO reports to suggest that this under the non-starved condition or normal conditions, the HUR is actually present in the nucleus. It cannot see the microRNA repressed messages, and the microRNA is actually dominating to repress the target genes. But under the starvation condition, when you need the expression of certain stress response gene, which is under microRNA control, they are binding the target mRNA, which have the each UR binding sites. And by the process, they also replace the microRNA from the Argonaute proteins. And this microRNA is now bound to the HUR, traffic to the endoplasmic, from endoplasmic reticulum to the early endosomes, where the protein get ubiquitinated. And this ubiquitination of HUR is actually responsible for its unbinding from the microRNA. And the microRNA get packaged into the endosomes and then ultimately exported out by the extracellular vesicles. So this kind of reversible binding and unbinding of microRNA by the HUR protein due to its ubiquitination under the starvation condition allows the microRNA packaging and export to occur. So that is the work we published in EMBO reports I mentioned, and it was a Human Frontiers funded project, so they highlighted the work. So next, we also explore the importance of this HUR-driven microRNA export in the other cell types, for example, in the macrophage, where we have found that HUR-mediated microRNA export like LET7 and 155 is actually responsible for the upregulated expression of TNF-alpha that happens during the LPS-mediated activation of the macrophage cells. Whereas the Leishmania, which is a pathogen which resides in the macrophage and try to suppress the immune response by reducing the TNF-alpha and other cytokine expression, pro-inflammatory cytokine expression. So they do so by targeting the HUR protein, and they cleave the HUR protein with the help of a GP63 metalloprotease that's secreted by the Leishmania, and that affect the HUR expressions and HUR level into the cells and affect the suppression of TNF-alpha expression. But if you overexpress the HUR or ectopically express the HUR in the macrophage, both in the in vitro as well as in the in vivo conditions, we can see there is a clear upregulation of cytokines that happens by the HUR, and that way it prevents the infection process by the Leishmania. So that paper we published in the EMBO Molecular Medicine to suggest that how the HUR microRNA exporter protein HUR is able to clear the internalized pathogen. So the next questions come in our mind, how the microRNA are packaged into the exosomes. So we already identified the HUR and its ubiquitination, how it affects the microRNA loading into the process. But to understand the mechanism in more detail, how exactly the microRNA are packaged, what are the molecular players present on the endosomes that play a role in this kind of HUR-driven microRNA import process, or which kind of molecule may act as an inhibitor of this process, we want to develop an in vitro system. Because without this in vitro system, it is very difficult to understand the mechanistic detail of any biological process in that sense. So we isolated the endosomes that would be the starting point to do this kind of in vitro assay development. And we do so by using the post-nuclear lyset of a C6 cells on irational gradient to separate the organelles based on their density and collect the fractions, analyze the protein marker proteins. So what we have done, we isolated the different fractions and analyzed on the Western blot to confirm their separation of endosome from the other organelles and polysomes. And then we analyze them again on a 5 to 15% irational gradient to separate the endosome depending on their maturation state from early to late. And we use that endosomes to confirm they are devoid of any ER contaminations and should be enriched for RAP5 and other kind of endosomal proteins. So with that, we started our reactions, and we also analyzed by the NTA how the size and the shape of the isolated endosome with atomic force microscopy. We also tested their size and shape. And then we use this endosome with the presence of ATP and incubate at 37 degrees centigrade in presence of the target substrate RNA. And after the reactions of 15 minutes, we digest out the extra microRNA present in the solutions. Only the imported microRNA that are protected from the RNAs should be to be recovered after the centrifugations, and we analyze the content. And when we do so, we find that there is a time-dependent increase of the incorporation of microRNA 122 as a substrate happened with the endosomes isolated. And this C6 cell doesn't express the microRNA 122. So all the microRNA we observed here is actually derived from the substrate microRNA that are present here. And we also did this kind of same experiment with a time-dependent way, and we find that 37 degree you have an increased level. And if you use a deoxycholate after the reaction before the RNA's digestion to lyse the membrane and find that the RNA is no more protected by from the RNAs after the import. So it suggests that the imported RNA is actually getting protected from RNAs and not either way. So we also wanted to check the dependence of energy for this process, and we do the experiment in presence of different concentration of ATP to detect that with increasing concentration of ATP, we find there is an increase of import occurs, and even at 1 millimolar ATP. And if ATP is replaced with AMP-PNP, which is a non-hydrolyzable analog of ATP, we cannot see the import process to occur. And at the same time, if we supplement it, that it is getting back to the normal level. And if we treat that with the apide to digest out the ATP present into the system, we can see there is a drop of the import process. So it suggests that it's an ATP-dependent process. So how the RNA substrate can affect the process? The microRNA we provided for as a substrate is a single-stranded form, but we can provide them in other forms, like in a double-stranded, partially double-stranded form with a miRNA, with an antisense completely base paired way, or using a 2-methyl oligo microRNA complex. The 2-methyl oligo microRNA has a higher affinity to bind to each other, and that's why it has a stable double-stranded form. And we find that with the antisense, we have an increase in the import process, which is also happening with the substrate mRNA. But if you use an anti-2-O-methyl, like 2-O-methyl modified, there's a drastic drop of the export that suggests that only the double-stranded form is getting recognized possibly for the import process. But before the import, the double-stranded need to get uncoupled from the microRNA, and only the single-stranded form get incorporated into the endosomes. So this is the way we find that this process is, however, the substrate specificity. And we also want to check the pattern of this import, whether this depends on the HUR protein, because we already identified HUR is one of the important candidate for the import process to occur. So what we did add recombinant HUR, which is pectinified from the bacteria, to the in vitro reactions, and we carried the reaction in presence and absence of the recombinant HUR protein to find that with increasing concentration, there is an increase in the import process. It actually happens very strongly at 500 nanomolar of HUR. The import process very much get increased, and the process get affected if you use an anti-HUR antibody to reduce the import process. And if the HUR is not functional by the use of the antibody, you have a drop of the import process. So that is interesting to see that HUR is important, but how is HUR doing the process? We find the process also depends on the GTP. So if we have a GTP in the reaction, you can see an increase in the import process, which is not happening in the presence of non-hydrolyzable analog of GTP. So you can see a drop of the import process. But when you draw, there must be a GTP is therefore involved in this process. And when we do an IP reaction with the HHUR, and we find that Raleigh is a protein that is coming down with the HUR in the IP reactions, and Raleigh is known to have a GTPase functions. And when we use the Raleigh to express in the cells, we find there is an export of microRNA get increased for different microRNA by the expression of Raleigh. And if we use the Raleigh expressing cell endosome, we find there is an increase of import process happening with the overexpression of Raleigh. And this Raleigh is also present on the endosome by the western blot, we confirm. But if we use an anti-Raleigh antibody, we can, like the HUR, we can see a drop of the import process. But even with the FH Raleigh expressing cells, we cannot see a increase in the import process when you use an anti-HUR antibody. So that suggests that HUR acts upstream of Raleigh. And if we do an GTPase activity assay with the purified Raleigh in presence and absence of HUR and microRNA, we can see only the Raleigh microRNA Raleigh having an increased activity of GTPase in the presence of recombinant HUR and microRNA 1 to 2 complex. So in nutshell, we are in a can suggest a model where the HUR microRNA complex interacting with the Raleigh, helping the GTPase activity, and in that way the HUR getting uncoupled from the microRNA, and then microRNA get incorporated into the endosomes for the export to occur in the subsequent step. So in the HUR may be recycled in the process. So we have identified the GTPase activity of Raleigh is increased by the interaction with the microRNA boundary recombinant HUR. So this is the first kind of mechanistic angle that we have been explored, how the HUR driven export may happen with the help of a protein present on the endosomal membrane. So you can imagine this is a nice system to start with, exploring different kind of inhibitors that may affect this kind of import process. We can talk about different viral proteins like TAD, ENV, and others that may affect the input process of microRNA, and by that way affecting the cellular processes in the infected as well as in the neighboring cells. So how this kind of transferred microRNA become effective in the recipient cell is the next question that I'm going to address. So one can imagine that microRNA are exported via the exosomes and they are released into the intracellular space, and they must targeting some of the target cells where they become functional. So there are a few questions that we don't know the answer on. For example, how the EVs are getting taken to the target cell, whether the endosomal pathways are involved in this uptake process, how they escape the lysosomal pathway, and how the microRNA that packed in the endosome get released and become functional in the target cells. So that are the questions that we are targeting. And then Battacharyya did all the experiment as a PhD student at that time. So we use a very simple system. We use a HeLa cells, which doesn't express the microRNA 122, which is a liver-specific microRNA. It also expresses CD63GAP for doing the microscopic analysis of the transfer process to occur. So after growing the cells, we can isolate the EVs and do all kind of biophysical parameter tests like NT and AFM and Western blot to confirm the EV is actually produced. And we can measure the miR122 content there also about the things. By following microscopically, we can see the visualization how the CD63GAP containing EVs are getting targeted to the other target cell which is not expressing the CD63GAP. And we can use also the EVs to treat into the recipient cell in case a HeLa cell that doesn't expressing the miR122, and follow the amount of miR122 transfer by measuring its content as well as the repression activity. For the repression activity, we use a Renilla reporter with microRNA binding site, perfect binding site, to see the level of change, activity change that happened after the EV treatment. So we find that with the EV treatment, you have an increase in relative level of microRNA 122 in a time-dependent manner. And also you find that it's happening for the miR122 also, but if you use a miR146 for instead of miR122, you also can see similar kind of transfer. So this is not a microRNA specific event that we are already following. It can happen for other microRNA as well. But for the sake of time, we concentrate on the miR122 itself to see that the repression level also goes up when you transfer this microRNA. Its endogenous targets of the miR122 in the recipient cell also goes down, and it doesn't affect the other kind of target for other kind of microRNA like KRAS by LET7 and so. So transferred microRNA are functional in the recipient cells, and we want to check whether the microRNA that transferred to the recipient cell is getting attached to the Argonaute protein present into the recipient cells. What we have done, we express the flag AGO2 into the recipient cells and do the co-culture together with the miR122 expressing HeLa cells, and to find whether these AGO2 that are present in the recipient cell get incorporated the Argonaute, the microRNA transfer there, and we find that AGO2 associated microRNA level goes up after the treatment, and it's actually also reducing the association with the target genes, in that sense confirming its functionality, and it also suggests the repression also occurs very much after the transfer. So that is the model we have adopted. But when you're analyzing the microRNA level, we have to understand that there are other players that play a role in the microRNA incorporation into the Argonaute protein for the functional microRNP2 form. So TRBP, dicer are the two proteins that are involved in this protein complex formation for the transfer of microRNA to the Argonaute to form the functional microRNPs. And when we find that AGO-associated microRNA level, it goes up against with time. But if we do the siRNA AGO2 treatment, you can see the reduction happens. So it depends on the Argonaute 2 protein. The microRNA can be transferred to the other Argonaute protein also, like AGO2 and AGO3. But if we deplete the dicer protein into the system, which is responsible for the microRNA, double-stranded microRNA processing and loading, we don't find any effect it has on the EV-derived microRNA incorporation into the Argonaute protein. So the process is dicer independent, although endogenous microRNA production get hampered by the depletion of dicer, but not the EV-derived microRNA loading to dicer. So how the endocytic pathway is involved in this internalization process? We did the experiment in presence of different siRNA against the F5A, F7A, or RILP, that are the component that responds for the endosome maturations and present on the endosomes early, late, or lysosome. So they are all important protein for the endosome maturation process, and we find that with that depletion of those factors, we can see an relative increase of microRNP level occurs, although the repression level goes down. Although the microRNA level goes up into this depleted system, the endogenous activity goes down, although the ago association goes up. So this was a little bit confusing, and we do that experiment to find that endosome is the sites where the accumulated microRNA actually present with the ago2 with the endosome, not in the lysosome or the ER. And with other kind of experiment, we find to conclude that this process is actually happening through the endosome maturation process. What happens, this miR122 containing extracellular vesicles was taken up by the endocytotic process, and these double-stranded structures, double membrane structure actually formed, and this endosomal membrane then fuses with the plasma membrane to release the microRNA content, and is driven by a pH-dependent fusion of the internalized EV with the endosomal membrane and release of the microRNA that happens. And this microRNA is a single-stranded form, is now getting loaded to the Argonaute protein present on the surface of the late endosomes that are also microRNA unloaded. We had other publication to suggest that there is a fraction of Argonaute always present on the MVB and late endosomes that are without microRNA. And these Argonaute now get loaded with the microRNA without the help of this dicer protein. It doesn't need the dicer to load it. And this process is very much dependent on the pH change that happened during the endosome maturation. If you block the process by bacillomycin and other kind of hydrogen pump blocker, we can restrict this kind of lysosomal maturation process and endosome maturation process and the microRNA loading to Argonaute. So these Argonaute microRNA complex, once formed, they go targeted to the endoplasmic reticulum, where they find the target mRNA to repress. So this is in nutshell how the internalized microRNA are getting activated into the recipient cell, and one of the first report to suggest the whole mechanism of this microRNA internalization. So these are the findings that I already discussed that are suggesting this process. So Battacharyya did almost all experiments and by herself, and she published this paper in this non-canonical Argonaute loading because it doesn't require dicer of extracellular vesicle derived exogenously single-stranded microRNA in the recipient cells in GSES. But one question that still remains unanswered is how do EV transfer to the matrix? We already understood a little bit about how the EV are generated with the microRNA, how EV get taken up into the target cells, but we are not sure till how EV get transferred to the matrix. So recently we have one paper published in JBC to suggest during this high lipid exposure, the export of microRNA 122 happens from the liver cells along with the export of dicer. So this accelerated export of dicer in the lipid-channel hepatocytes actually buffers the cellular microRNA level and prevents cell death. And this miR122 is actually an inflammatory microRNA because it can go to the other tissues or liver-resident macrophage to activate them. So we want to check whether this kind of transfer of the microRNA 122 containing EVs can be stopped. So what we have find that there this kind of transfer of EV from hepatocytes to the recipient cell present here can be studied in this two-chamber cell culture model, where you put the EVs with the microRNA into the upper chamber separated by a membrane, and in the lower chamber you have the cells grown on the surface, and you can measure the transfer kinetics. So you can see that the EV we have isolated and with a CY3 miR122 present there, and we can follow them microscopically to the transfer here, or the CD63 GFP as well. In both cases, you can see the process get retarded heavily by a chemical called ARP101. ARP101 is an inhibitor of the MMP2 family of matrix metalloprotease. And if we use a recombinant matrix metalloprotease, you can see there is an increase of the process happens. And this process is also getting affected if you have EVs secreted by the MMP2 depleted cells, which are treated with the siRNA against the MMP2. So it suggests that the MMP2 is also present on the EV surface, and you can do this from this Western blot, you can understand that. So it happens that MMP2 inhibitor 101 can prevent this EV transfer to the target cells. we want to see the effect it has on the target cell transfer. In case of recipient macrophage cells, we can see the transfer of this Mi122 in presence of Mi122 expressing cells. The transfer of Mi122 is very much synchronized with the expression of TNF alpha or IL-1 beta, which is getting affected by the ERP101 and getting positively influenced by the recombinant RMMP2. So that suggests that MMP2 is required for the hepatic Mi122 containing EV transfer across the extracellular matrix to the macrophage, because we go the matricial here to prevent a matrix kind of situation to see that its effect on the transfer process. And if we use the EV prepared from these Mi122 expressing hepatic cells and treated them with ERP101 or without, and then inject them into the mice to see the effect it has, we can see the transfer of miRNA1 to the liver resident macrophage get heavily retarded in presence of ARP101, and which is also related to the IL-1 beta expression, TNF alpha expression, IL-6 expression in those cells. So that suggests it is an interesting mechanism how this MMP2 play a role in allowing the EV transfer across the matrix. So EV is recruited by the hepatic cells with the MMP2 will able to transfer to the macrophage resident there, because it digests the collagens and other matrix proteins to allow this passage through to the target cells. But if we use an MMP2 inhibitor and see that this kind of transfer is not occurring. So as we have already identified this high fat or NASH condition, there is an export of miRNA122 containing EVs from hepatic cells to the macrophage for their activation to occur. So one can think about to develop this kind of inhibitor to stop this process and restrict this kind of metaflammation related problem. So this EV is not only secreted by the higher eukaryotic cells, it can be secreted by the pathogen itself. For example, GP63 is a protein that's secreted by the Leishmania, which is present on the EVs, secreted by the Leishmania, and that can cleave the dicer protein in the hepatic cell to prevent the microRNA production there, to reduce the cholesterol production in the infected animals model of Leishmania donovani. So that was one of the examples that we have documented earlier, how this process can be universal. In the same context, we can find that Leishmania donovani infected cell also secrete EVs with the miR146 containing microRNA, which is an anti-inflammatory microRNA. By going to the naive macrophage, it polarizes them to the more M2 state by reducing TNF alpha production and IL-1 beta productions and inducing IL-10 there. On the other hand, in the hepatic cells, miR122 production is hampered by miR146, but miR122, when secreted, it can activate the macrophage to more M1 macrophage kind of situation. So can we use miR122 or miR155 containing EVs for immunomodulation of the T cells? That may be a useful aspect in the context of HIV infections that is interesting to explore on. So with that note, I like to end, and I like to acknowledge all the help of my colleagues. Most of them are the PhD student who did a lot of works. I presented the work today primarily done by Shobhi, Komalika, Sudarshana, Devduti, Ornov, and Bortika. I didn't have chance to discuss the work done by the others, and maybe in another platform I will acknowledge them, their work. And our funding was very generous. I received most of the funding from Human Frontiers and Wellcome Trust and other funding agencies. In India, they are very generous to support the research, like Department of Biotechnology, Department of Science, and some bilateral program like CEFIPRA with the France, and we had, and also in Switzerland, we have a lot of project funded. And right now, I am using the startup fund from UNMC, and I like to acknowledge Dr. Howard Gentleman for his vision, for his enthusiasm to recruit me and into this department as a professor. And I would like to explore the EVs into context of neurodegenerations and also in the context of HIV-mediated HIV infection and eradication and role of the EV in this process. Thank you all for your kind attention, and I'd like to answer all the questions you may have now. Thank you.
Thank you, Dr. Battacharyya. Dr. Gendelman, would you like to unmute and ask him the question? I think you're still on mute.
I'm sorry. I usually have a big mouth, but your pants, when it comes to the internet, you're restricted. First of all, I came in a little late, so I apologize for that. But I wanted very much for Dr. Battacharyya to present, because we've done something very unique for the department and our community. And I thought it's critical for everyone to see that not only is he a great scientist, there's no question his publications and his work comes before this presentation, but also to welcome him to our community in neuroimmunology, neuro-HIV, and neuroinflammatory disorders. One of the big issues, and I think Fata will help me with this, ask a question if he's still on, is that EVs are still in its infancy in terms of therapy. And maybe you agree or maybe you disagree that we're much further along than what I'm saying, but I truly believe that we're still in the first or second stage or inning of this 9-inning game in the World Series of EVs. And I think the big issue, which is in terms of microRNAs, is delivery, is using EVs as a delivery system for therapeutics. And we're hoping as we get closer to HIV elimination and excision and immunomodulation, that EVs will continue to play a very, very important and expanding role. And we're very proud to be able to have recruited Suvendra into our program, and again, to welcome him to this community. So I'll stop there and let others open this up for discussion.
Thank you.
Thank you, gentlemen, for your kind support.
Yeah, Howard, I would echo we might not even be in the World Series yet with the EVs. But anyway, I agree with your comments. I've really enjoyed your presentation, and just had a quick question on about the EV transfer across the extracellular matrix. You talked about the microRNAs needing EVs, but are there other mechanisms that these microRNAs can be transferred, like through ribonuclear protein complexes or lipoproteins, or even naked? Is that occurring? And if so, what the percentage is estimated versus EVs?
Yeah, I think that's a very nice question, a very important question as well. The microRNA can be detected in a naked form or is in complex with the Argonaute protein into the blood or serum that people have detected. But we cannot be sure they are there because of some degradation product of the EVs that are lysed during this transfer process in the blood, and they are there because of that. And they are also taken from some kind of microvesicles which are bigger in size and released in a different mechanism than the EVs or exosome rather, and that can cause a effect also in the target cells. So in the targeting of this microRNA in a different form to the recipient cell is important, but in the same time, it may have a different mechanism for the uptake. I'm not ruling out it. They may have a different mechanism of this kind of uptake and transport as well. But preliminary, people find that EV is more important, because if you stop this kind of EV production or EV incorporation of specific microRNA, you can see the physiological effect. So that's why people are more concentrated. Although there is a debate of how much microRNA is actually present in EV compared to the other RNA component present in EVs. But again, we have to consider that EV is very much in heterogeneous in compositions. They may have specific kinds of EVs that are specifically selected for the microRNA to export, and they may have a different kind of biological function compared to the other EVs, which may have other cargoes to carry. So as Dr. Gentleman mentioned, this field is still in infancy, not only in the context of delivery or use it as a biomolecular tool, but in the context of its understanding. So I'd like to play a major role in both aspects, like how these microRNA are getting trafficked, how does the biological role of this kind of EVs and therapeutic angle, but also understanding the fundamentals of how the process happens. And development of this in vitro system is one of the way to address this.
Great, thank you. Thanks.
Along those lines, Dr. Battacharyya, I had one question. We do some drug delivery with nanoparticles and other things. EV is a much more complex, like you said, heterogeneous system. What are the biggest challenges if we, in general, is it more of isolation, or is it more of loading, or just the complex nature? And what are the advantages over the other systems that are similar, like liposomes and nanoparticles, of using EVs, especially for biologics like microRNAs compared to small molecules? I'd like to.
I think that's a very important question to address, and I think one of the important aspect that we have to consider in this context of EV is that is low immunogenicity. So as they derive from our body and it can be generated from the cell line you are trying to get it. For example, you can isolate some kind of immune cell isolated from the body and allow them to secrete EVs with the specific cargoes in it and use them in future. So that's the target. On the other side, that there are some biological fluids like milk have a lot of EVs in it. And one can think about to produce EVs in bulk from those kind of biological fluids that can be used in a low-cost manner compared to any kind of nanoparticles. There is a difficulties to sometimes to load the cargoes into the EVs. I think a lot of chemical approaches have been there in place to load specific microRNA or proteins into the EVs by different methods. But one of the easiest way is to use the cells to express the specific microRNA into it and packaged into it with the help of some RNA binding protein like HUR we are talking about. If we express the HUR and microRNA together, maybe they will help to get the microRNA packaged into it. So that's why you can get EVs with a high concentration of specific microRNA and use them with low immunogenicity to inject. And their biodistribution is also very important because it can easily reach brain. For example, if you isolate EV from the macrophage, they have the intrinsic capacity to reach to the brain. So that are all advantages in the context of other kind of nanoparticles like lipid nanoparticles or liposome.
Thank you. I see there are a couple of other. Please unmute yourself and ask the question.
All right. Great talk as always, Suvendra. My question is, when they are released in our physiological system under pathology or whatever, these EVs, do they all carry homogeneous cargoes? Are there heterogeneity? Is some cell taking it? Specificity? Can you comment on that, please?
Yeah, I think that's a nice question, and it's actually follow up with the comment I had previously on that it is a heterogeneous system. So EVs definitely is a heterogeneous system, and people are trying to address the heterogeneity by isolating specific EVs or single EVs to identify the cargoes present in it. So maybe it's the decoration of the EV's membrane by the different proteins and on the surface is actually determining its targetability. So it may targeting specific cells or allow it to cross specific blood-brain barrier or other kind of boundaries because of the presence of this kind of EV surface markers. So that allow them the specificity. And the cargo selectivity, I think it also depends on the context. If you isolate EV from one context and to the other, for example, starvation versus normal, you may have a different sets of microRNA and protein present there, and that's actually allow the targeting to the specific kinds of cells. Maybe it happens that under the stress condition, you have the EVs that have the highest level of microRNA 1 to 2 present from the liver, and that may be targeting to the other peripheral tissues to induce inflammations there in the heart and other peripheral vascular structures. So that's some way it's a crosstalk that actually context dependent, and the cargo is also very much context dependent. That's my assumption.
Thank you.
Dr. Battacharyya, thanks. A great talk again. I think it's a lot of potentials open. I'm just wondering, based on all these questions that came out, the heterogeneity, targeting, and you illustrated very well. There's any examples in the literature or anybody has done in different contexts or any disease or whatever targeting microRNAs could be potential therapeutic applications in the future. Could you illustrate a bit on that?
Yeah, the targeting microRNA has already been tested in not only context of the EV actually, but in the context of miR1 that I'm talking about. When you're talking about the druggability of the microRNA targeting is there, yes, in the hepatitis C virus context, People have tried to use the antisense oligos of the 2-methyl oligos and other kind of oligos, LNA modified oligos, to use the inhibition of miR-122 in the liver, because miR-122 is used by the hepatic C virus for these applications. So I think that clinical trial has been in the very late phase to as a drug already probably is there. And a few other microRNA present in the retina and others, they are also getting targeted. People are targeting specific microRNA for the cancer as well. LAT7 is one of the interesting target people are using for the small cell carcinoma or small cell lung carcinoma. So this is also interesting candidates. People trying other microRNA in the pancreatic cancer context. And when you're talking about the EVs, I think people are using the specific microRNA to target the cancer cells, derived EVs, to test their effect on the cancer metastasis. So I think there are quite a good number of clinical trials going on with microRNA and as well as with EVs.
Oh, okay, that's great. Yeah, so now I think the approach you are trying to develop to target the CNS diseases, I believe, right? That's where a little harder.
Yes, that's one of the primary
barrier and things like that. Okay, got you. Thank you.
Thank you.
And any other questions? Okay, we'd like to thank our speaker again.
Wait, one more question.
Sorry about that.
One more question,
Yeah.
if I can. I put this T-shirt together just for Suvendra's talk. Suvendra knows what this T-shirt is, but we started a collaboration for extracellular vesicles on a farm in Nebraska. And he's laughing because he's spent many, many hours at the farm. But what we found is that from colostrum, a newborn calf milk, we can generate 10 to the 14th, a tremendous amount of EVs.
Yes.
And what happened is we had a big party. Suvendra, you were at the party just a few nights ago with a band and dancing to celebrate the farm. And obviously the owner of that farm, who's being a big supporter of this project. But now we're all wearing T-shirts to celebrate extracellular vesicles acquisition for his new research enterprise in Nebraska. So we're going to be giving this out. If people come to the SNP meeting, the first 5 best posters on EVs will get a T-shirt. I just want to let people know about that. Thank you.
Thank you. And anything else to add there?
Yeah.
All right.
I just like to add that I like to thank everybody who present here and so interested into the research we are doing or have been doing. And I like to convey my personal thanks from me and my wife Kamalika, who is also very much part of this project and doing as an assistant professor work on this milk EV here, as the gentleman mentioned. So for our recruitment and on tremendous support that the gentleman is providing to us, his insight and all the vision he has, we'd like to work very hard on that to make it possible.
Thank you. And thank you everyone for joining today.
Thank you. Really enjoyed it. Nice job. Nice job.
Thank you so much. Next week, we have Dr. Lily Brown presenting. I don't have her title yet, but I'll be sending that out shortly. We will see you all next week.
Thank you.
Thank you.